For the FAFO methods, because the FAFO methods considers the PCR efficiency of the targets and the reference gene, in the formula for calculating the gene expression, you need to calculate these efficiencies first. Assuming PCR efficiency of target gene is 93% and PCR efficiency of reference gene is 101%. In the FAFO method, it is assumed that an efficiency of 100% has a value of 2. The PCR efficiency conversion, or we call PEC, is to use percentage of primary efficiency divided by 100 and then plus 1. So for the target gene, it's 93% for the PCR efficiency. You will use 93 divided by 100 and then plus 1. You will get 1.93. That's your PCR efficiency in the FAFO methods. For the reference gene, you have the PCR efficiency of 101, and then you use this value to divide it by 100, and then followed by plus 1 of the value. Then you will get 2.01. That's the reference gene of the PCR efficiency. The delta CT here will be use the control average to minus average CT. So you will have the 17.18, 16.96, and the 17.11 for control group, and 18 and 17.95 and 17.88 for the target group. Same thing applied to gene of interest. So the average of control will be 17.08, and the average CT of the gene of interest will be 13.63 in this case. So the gene expression ratio would be used, your target PCR efficiency of this number, which is 1.93 here, to the delta CT of gene of interest. And then this value will divide it by the PCR efficiency of the control, PCR primary efficiency, which is 2.01 in this case, powers to delta CT of housekeeping gene. Then you will get the ratio of the value for each of the control replicates and the treated replicates. In this case, for control 1, 2, and 3, you will get the ratio 1.13, 0.97, and 0.92. For the treated group, you will get the fold of the ratio 44.62, 49.15, and 45. Here, I just want to show you that in our previous publication, we also doing the same thing by using real-time PCR to measure in Nile tilapia, or we call Aureochromis niloticus. When we treat the fish with the extract of microalgae Sarcodia sui, we see the increase of genes, of several different immune genes. And then after that, we see that the increase of these immune genes will help Nile tilapia against the insult of Streptococcus agalatia, which is a gram-positive bacteria, notorious in aquaculture. We first test which extraction methods, or pressured liquid extraction or solid liquid extraction method, will allow us to obtain extracts that can stimulate immune responses of cells effectively using real-time PCR as a way for readout. That is, we use different extraction methods by pressured or autoclave extraction method, or use solid liquid extraction method. After that, we obtain the extracts. We put those extracts in the cell medium and we treat the primary cells or we treat the cell lines, and then we want to see whether the response would be the same or different at different concentrations of the extract, and also for the different time points. And the readout will be real-time PCR in this case. As you can see here, we will always get higher expression of those immune gene response after we treat the cell with solid liquid extraction extracts. And then on the right-hand side, you can see here, after given the fish of the extracts, and then we challenge the fish with Streptococcus agalatia, which is the pathogens for the fish. And we observe the survival rate of the fish compared to the control group without giving the extract. After observation, we see that higher survival rate exists for the fish that injected of the extract that we obtained from the solid liquid extraction methods. So here we see that we can use real-time PCR to quickly monitor the expression of the genes of your interest, and then you can translate those immune responses into a real world of the aquaculture.